Let's continue what I was explaining last time. When the time ran out, I was explaining the blood vessels, and I explained already the arteries, veins, and the capillaries. I just wanted to mention that the capillaries, they deliver oxygen and nutrients to the cells and take away carbon dioxide and waste products. And also, the blood is always carrying these nutrients, gases, wastes, hormones, antibodies, whatever. The presence of the amounts of nutrients and oxygen together with wastes in the cells, in the blood, will maintain the concentration gradient, so it is easy for these substances to diffuse in and out of the cell. Now, there are certain arteries or artery names and vein names that you have to know. The blood is returned back to the heart by certain veins. The first veins that you have to know that deliver the blood from the lungs to the heart. These are called the pulmonary veins. And there are other veins that deliver blood from the liver to the heart. But what's the name of this vein, Salman, that delivers the blood from the liver to join the inferior vena cava and then to the heart? You remember its name?
I don't remember. I don't remember.
It's the hepatic vein.
I don't remember the hepatic vein.
I mentioned it when I was explaining the digestive system. I told you that if this is the liver, for example, and all the blood from the abdomen is collected in the hepatic portal vein, and then this hepatic portal vein is going to enter the liver and branch extensively inside the liver in order to filter the blood from toxins and from excessive amino acids, amine groups, to form the deamination. And then these veins will collect again to give us the hepatic vein, which is the one I just asked you about. And this hepatic vein is going to drain into the inferior vena cava, and this will drain into the right atrium of the heart. There is a difference between the hepatic vein and the hepatic portal vein. Portal vein means something deal like a port, la porte, the door طبعا in French. فالhepatic portal vein, it gives the blood to the liver, where the liver filters some materials of the blood, takes away excess materials, and then the blood returns back through the hepatic vein. The third vein that you have to know is getting blood from the kidneys to the heart. You remember this name, this vein that delivers blood from the kidneys to the heart?
Inferior vena cava.
At the beginning, this vein, yes, it pours in the inferior vena cava. Here I have the inferior vena cava, and here is this vein that's coming out of the kidney. This is called the renal vein, and it drains inside the inferior vena cava. And then the inferior vena cava will collect the rest blood from the body and deliver it to the right side of the heart or the right atrium. Because you are asked about the names of such veins, where sometimes in a drawing they ask you to point to a certain vein. That's why you have to know the names. But what about the arteries? The important arteries that you have to know. Of course, the arteries will deliver blood from the heart. If the artery delivers blood to the lungs, then this is, of course, the pulmonary artery. And as I said before, that it is an artery that carries deoxygenated blood and not oxygenated. Also, from the heart to the liver, the same name. And
Hepatic artery.
yes, it is the hepatic artery. And this is, by the way, the artery that carries the waste. So sorry, this is the liver, not the kidney. Hepatic artery. And from the heart to the
Renal
kidney. Yes, very good. To the kidney, it's the renal artery. And here is the artery that carries the waste from all the body to the kidney in order to filter the blood, of course, as we will see in the excretion part. There is another part, or other kinds of channels or vessels inside our body that are called the lymphatic vessels, and they are a part of the lymphatic system. So what is this lymphatic system? This is a system of connected channels, or looking exactly like the blood vessels, but with slight differences, that form the lymphatic system. So the lymphatic system is formed of lymphatic vessels together with very important nodes that are called the lymph nodes. So what are these vessels and what are these nodes? The lymphatic vessels, they run parallel to the blood vessels in close contact with them, and they are responsible for collecting the tissue fluid. I'm going to say now what is the tissue fluid, of course. And about the lymph nodes, they are responsible to store and to release white blood cells of a certain type, which are called the lymphocytes. Of course, the lymph nodes, where they are storing the lymphocytes, and when there is an infection or whatever, so these white blood cells are released in order to defend the body against the pathogen or the microorganism or any foreign body that entered the body. But about this tissue fluid, what is it formed of? Now, if you can look at this picture, this is the one. Now here, this is a picture for the blood capillaries. As you can see, these are the blood capillaries and they are full of blood. Here are the red blood cells, of course, and this is plasma. El plasma in the capillaries, it gets out to the space in between the capillaries and the cells. So if this is a cell, so there is a space between the blood capillaries that through which to which the plasma escapes out of the wall of the capillaries. As you can see, that the wall of the capillaries has holes. The plasma is going to escape outside these holes in order to get rid of some materials that will form something called the tissue fluid. This tissue fluid is a fluid that's found in between the capillaries and the cells, and they are nothing but plasma. The plasma, it gets rid of some materials to this tissue fluid, and then again this tissue fluid is returns, part of it returns back to the blood, part of it only, and the rest is collected by the lymphatic vessels. The lymphatic vessels, after collecting the tissue fluid, they drain their components into the heart again. يعني lymphatic vessels, they join a certain vein just below the shoulder, and then they deliver, here it is, let me show you this picture. Here, as you can see here, that all these are lymphatic vessels, and they are collecting the tissue fluid from every part of the body, where the material that is moving inside the lymphatic vessels is called the lymph. They collect all the lymph or the tissue fluids from all over the body and finally pour into a vein just below the shoulder. This is called the subclavian vein because it's written on the picture, this picture from your book. So the subclavian vein finally drains into the superior vena cava and into the right side of the right atrium. This means that the tissue fluid sooner or later is going to return back to the blood by the lymphatic vessels. The lymphatic system or the lymphatic vessels, as I just told you, that it has a certain function in circulating the tissue fluid all around the body, collecting tissue fluid, and also they produce or they release the lymphocytes which filter the lymph out of the body. As I told you now that the substances, they diffuse between the cells and the tissue fluids, and the lymph vessels are going to collect them back and return everything back to the blood. If you looked at this picture, another one for the tissue fluid or for the lymphatics, as you can see here, that this is the blood capillary, and tissue fluid is getting out of the blood capillaries like this. Part of the tissue fluid on the other side of the capillary is going to be reabsorbed back to the blood, while the rest is going to remain in between the cells and the capillaries to be collected by the lymphatic vessel. The lymphatic vessel, as you can see, the part here that is found in between the cells, we call it a blind lymphatic vessel because it's not opened from the end. No, it just collects and then returns back to the heart in one direction, of course. This picture is not found in the book, but I found it good for the students. Now, let me now go to the third part of the circulatory system, which is the blood, because I already explained the heart, blood vessels, and now the blood. Here, the blood, we say that this is the transport medium, or it is the medium that's going to carry everything, whether useful materials or waste materials to and from the cells. Blood is formed of two parts. It is formed of a liquid part, which is called plasma, of course, and solid parts, which are red blood cells, white blood cells, and platelets. Sometimes the students consider the platelets as cells. No, they are not. They are just fragments of cells, or they are formed of different types of solid structure. Taban, the name, you don't have to know the name in OL, but this is explained in ES. Red blood cells and white blood cells, they are complete, they are normal cells, although the red blood cells has some modification that I'm going to explain now. About the function of the red blood cell, it carries the blood gases. So what are the blood gases? These are oxygen, of course, and carbon dioxide. Why? Because it contains, the red blood cells contain hemoglobin, and this is a blood pigment or a respiratory pigment that carries the blood gases that result from respiration of the cell, or result in and from. Yani oxygen will go to the cells to be used in respiration, while the carbon dioxide will act as a waste product to get out of the cell after respiration. The white blood cells, they are involved in defending the body. Defending against what? Against the pathogens or the organisms or the foreign body that cause diseases. So we have here two types of functions which are phagocytosis. It means engulfing, of course, of the foreign materials or the foreign bodies, and production of antibodies. These are the functions of the white blood cells, and both functions are for defending the body against microbes or against foreign materials. The platelets, they are responsible for clot formation or blood clotting. And of course, blood clotting is important to stop bleeding, and also it blocks the blood vessel so that it prevents the entry of microbes or bacteria from entering through an open vessel or a wounded vessel. These are the solid parts of the blood. But what about the plasma? Let me take the plasma in a separate board. Plasma, it forms most of the blood. Yani, if I have 100% volume of blood, all the blood volume, the plasma forms 54 to 56% of the total blood volume. This means that the others, they are going to form the rest, or 46 to 44% of the volume of the blood. This means that the plasma forms the majority of blood, and it is just a straw-like or straw-colored liquid that is responsible for transporting certain materials in the blood. What are these materials? The plasma transports ions or salts. It transports wastes. It transports hormones, antibodies, enzymes. Everything that can be transported will be transported by the plasma, except, of course, oxygen. Also, the plasma transports a part of the carbon dioxide. Yani always we say that plasma transports all the materials, but not carbon dioxide. Carbon dioxide is transported by red blood cells. No, yani a part of the carbon dioxide is transported in the plasma. I'd like here to mention one of the very important wastes, which is urea, because this is the mostly toxic kind of waste, and it is carried by the plasma, of course, in order to be released out of the body by the kidneys. Also, there is a very important component, which is the plasma proteins or the blood proteins. Blood proteins are very, very important to our life, where part of the plasma proteins, they share blood clotting. So what about the blood cells, and what about their structure and how do they look? First of all, the red blood cell. Red blood cells, they, excuse me?
Nothing.
Okay. The red blood cells, they were normal cells having a nucleus. And when the red blood cell is newly formed, we say that it is an immature red blood cell. It means that it cannot perform its function. Why? Because it contains a nucleus. Although the nucleus is very important to all the cells, but it's not important in the red blood cells. But if the red blood cell is newly formed, it contains a nucleus. Later on, it starts to develop and to differentiate, where it loses its nucleus. So the space of the nucleus becomes empty. That's why it looks like this, biconcave. If you looked at it from one side, you're going to see that as if it is compressed in the middle because of the removal of the nucleus, and the space of the nucleus is empty. If you looked from above or from below, you are going to see it like a discoid shaped cell, and in the middle as if it is compressed. Here we say that the red blood cell became mature or it can perform its function. But this is not enough. The removal of the nucleus is for a reason, because the space of the nucleus is going to be occupied by this red material, which is called hemoglobin. And once the hemoglobin is filling the red blood cell, now the red blood cell can perform its function, because this hemoglobin is going to trap or to hold the oxygen or to combine with the oxygen to give oxyhemoglobin, and it's going to trap part of the carbon dioxide to give carbaminohemoglobin. This is the red blood cells, and of course the red blood cell number is the majority of the cells in our blood are red blood cells. They are in millions. For the female, they are about from 5 to 5.5 millions of red blood cells in every millimeter cubed of blood. And for the females, these are the females. For the males, they are about 5.5 to 6 millions every millimeter cubed of blood. What about the white blood cells? The white blood cells, they are much larger than the red blood cells, and we have different types. We have large white blood cells that have inside a segmented nucleus. The nucleus looks like this, as if it is divided into segments. And these white blood cells, they are called the phagocytes. It means that they are going to phagocytose or to engulf the pathogens. Another type of white blood cells are smaller than these ones, and they have a rounded or nearly rounded nucleus inside. Looks like this. And these are called the lymphocytes, which are going to produce antibodies. So here, the lymphocytes will produce antibodies. And of course, you have to take care. These are not antibiotics. Antibodies, they are chemicals or proteins that are protein. They are protein in nature, I mean. They are produced from a certain type of lymphocyte, but in order to attack the bacteria or the pathogens. But the antibiotics, these are the drugs that we intake when we are sick. And phagocytes, they are going to make phagocytosis. So what is this phagocytosis? Phagocytosis means to engulf a foreign body. Suppose a foreign body entered and is found in the blood or whatever. Some of the phagocytes, they wander in between the cells in order to trap any foreign body that entered. Because phagocytes, they wander in the tissue fluid in between the cells. Here, the phagocyte is going to approach near a pathogen or a foreign body. And then this phagocyte is going to engulf or to make a shape like a gulf around this pathogen. Later on, this gulf is going to close more and more to include the pathogen, or to force the pathogen to enter inside the cell. Later on, after the pathogen is completely inside the cell, this sac or this vesicle is going to separate from the cell membrane of the red blood cell, because of course, white blood cell, this is a cell membrane, of course. And later on, the digestive enzymes are going to attack this pathogen in order to destroy it. This process is called phagocytosis, or sometimes we call it engulfing, but of course you have to know the scientific term, which is phagocytosis. So what about platelets? Platelets, they do not have a certain shape. They look like this, and they are very, very small, like this, looking as if fragments like this. They do not have a definite shape yet. Right. What about now? Let's go to the immune system. Or another chapter, which is chapter 10, the immune. Sorry, I forgot to explain the blood clotting. Blood clotting is a certain mechanism or a certain process that happens inside or in our blood when there is an injured artery or vein, and this will help, of course, to stop the bleeding and to block the injured blood vessel so bacteria cannot enter through and cause infection. So here we say that the blood clotting reduces the blood loss, or stops bleeding, of course, and it's going to keep the pathogens out by closing the blood vessel completely. Now here, when the blood vessel is injured, immediately the platelets, they are going to change their shape. Instead of having no definite shape like this, they are going to change their shape to become spiky. And this change in shape will, of course, attach, or as if it is clumping, it makes friction with the walls of with the cut wall of the blood vessel, and the platelet will start to release chemicals. This chemical, or the most important chemical here, is called thromboplastin. This thromboplastin, of course, will be released in the plasma, and in the plasma there are calcium ions already there, always there. But the thromboplastin, together with calcium ions and another substance, which is vitamin K, altogether they are going to activate an inactive enzyme that is found in plasma. This inactive enzyme is called prothrombin. So by these three substances, this prothrombin will be activated to give me an active enzyme, thrombin. So the difference between prothrombin and thrombin is that prothrombin is inactive, an inactive enzyme, and thrombin is an active enzyme. So here, this is thrombin, not thrombic. Thrombin. This thrombin is going to play a great role because it will activate a plasma protein which is called fibrinogen. This fibrinogen is soluble, of course, in plasma because it floats in plasma, and it will change by this enzyme thrombin into another insoluble protein which is called fibrin. This fibrin is going to form like a mesh, like a network like this, and the holes in between the network is going to be blocked by blood cells, platelets, the blood cells, of course, red blood cells, white blood cells, together with platelets. They are going to block all these holes in the mesh formed by fibrin, and this is, of course, the blood clot. So if you were asked, what's the blood clot made of? You're going to choose fibrin and blood cells. If you were asked how the blood clot is formed, then you're going to mention all this till the end. I would like to show you this picture for the blood clot. Mm, Here it is. Here it is. Now, if you can see that here is the injured blood vessel and platelets, of course, they are going at the end to clump here in order to block the blood vessel. But what is this clump? Here are the fibrin or the fibrin mesh, and here are the blood cells in between. Type, suppose they brought you this picture for the blood. This is called the blood film, or in other words, we say photo micrograph. Micrograph for a blood film. So what's the meaning of photo micrograph? It is a picture from light microscope. If the picture is electron photomicrograph, then it is from an electron microscope. As you can see here, that these are the red blood cells, these are the white blood cells, and these little dots are the platelets. Here is a hand-drawn picture for the red blood cells, white blood cells with their types, and these dots are the platelets. Now let's go to a next chapter, which is
I have a
about
question.
yes, of course.
Now you said when someone like gets injured, the blood clots, right?
Yes.
What would be the reason if someone got infected? Like blood clotting stops infection, right? But if someone got infected, what would be the reason?
Before the blood clotting, you mean?
Yeah.
Before the blood starts to clot. This means that it will be attacked, the pathogen will be attacked by the phagocytes, like this. Yani, the blood is always ready, and antibodies can be produced. Okay? Yani, if a pathogen gained access to the blood before the blood clotted, so there is a defense mechanism inside the body by defending the body by phagocytosis and production of antibodies to destroy completely this pathogen. Sometimes the pathogen is very aggressive, so it starts to form disease, and of course the antibodies are going to stand for it. They're going to destroy it completely.
But it takes time, right? Like you get sick.
Yes, they get sick, and it takes time for the antibodies to be released into the blood, of course.
But blood clotting is really fast, right? Like really fast.
Yes, yani, according to the diameter of the vessel injured. Yani, if you are
the larger, the longer.
If the diameter is narrow, so it's going to clot very fast. Yani, suppose you were pinpricked, a pin pricked you. Blood is going to get out, and after few seconds, it's going to stop because the blood clot was formed in this capillary. But if, of course, the diameter of the blood vessel is more than the diameter of the capillary, so it's going to take some time. That's why we have to cover it, to cover the wound until the blood clots. We usually use gauze or a plaster or whatever in order to cover the wound, to stop the entry of pathogens and to give a chance for the blood to clot without any entry of pathogens. طبعا, if it is not covered, so it's going to be contaminated, and bacteria are going to enter.
Okay, because I never cover it.
لا, you have to cover
I never ever cover it. Like maybe after an hour, then I will cover.
You have to cover it if the wound is not very, it's not a small wound. You have to cover it. After the blood clot is formed, if this is the vessel, the skin, and here there is the wounded blood vessel and the clot was formed, above the skin there is something called the scab. You must not scratch it, because I know that everybody
I know that.
scratches the scab, of course. You have to give a chance also for the skin to reform again. But
And
when
that's why it starts to like going itself. هذا
excuse me,
يتقشر ولا هيك شيء يعني.
yes, of course.
Okay.
يعني I know that everybody, including me, when I was young, we used to scratch this scab. It doesn't look good. But the skin is not reformed yet. We are extending the time for reforming a healthy skin. But of course, we don't know. When we are young, we don't know about this.
So like the scab is to protect the skin from infection, like for the bacteria to not
Yes, of
land
course.
when it's repairing.
Because you know that the skin, when you are injured, the skin is open. It's not only the blood vessel, it's the skin as well. We have to give enough time for the skin cells to divide, to form mitosis, or to form new cells. This scab protects the skin until it is reformed again, or repaired, in other words. But we do not give the skin a time. We only scratch it. Let's go now to chapter 10, which is about diseases and immunity. First of all, I have to mention that the disease is a certain disorder that is caused by a certain pathogen. Now, what's meant by pathogen? Pathogen is a causative agent to diseases. And we have different types. We have bacteria, we have viruses, we have protoctista, and we have, of course, fungi. So any one of these can be a pathogen that leads to the occurrence of disease. There are certain types of diseases that can be transferred from one person to another, and these are called transmissible diseases. Like what? For example, like even influenza, common cold, TB, whatever. These are diseases that can be transferred from a patient to a healthy person. But there are some diseases that are not transmissible, of course. So, like, for example, like cancer. It cannot be transmitted from one person to another. Transmissible diseases, these are diseases in which the pathogens can pass from the patient to a healthy person. And these are our concern, of course, not the non-transmissible diseases. The pathogen for a transmissible disease may be transmitted in either a direct form, or we call it the direct transmission or direct contact, or indirectly transmitted. What's meant by direct contact? Direct contact, for example, means through, for example, the blood, or through tissue, or through the fluids of the body. Indirectly, it means that if there is a contaminated, for example, food, or polluted food, polluted water, also from the air, droplets. This is indirect. Here, direct contact means that the blood of the patient or the fluid of the patient must come in contact with the blood or fluids of the healthy person. For example, if there is blood transfusion, or there is somebody that needs blood, so they insert blood or inject blood in the body of a healthy person. If the inserted blood or injected blood is contaminated with bacteria, so the other person is going to catch a disease, because the blood, the unclean blood or the infected blood is going to come in contact with the blood of the healthy person. لكن الدروبlet or the airborne infection, waterborne, foodborne, it means that the bacteria are found in certain substances and these substances are taken inside our body. Food and water, of course, if they are contaminated with a pathogen, so they can enter through ingestion or by eating or drinking. Air is inhaled. That's why we say droplet infection. If somebody is having, for example, a flu, influenza, or whatever, and they started coughing, so with coughing, droplets coming out of the mouth, droplets of saliva, of course, they get out of the mouth together with the bacteria. So if somebody's standing here, they might inhale these bacteria from the droplets. This is an indirect way in which the air is carrying the pathogen from a patient to a healthy person. That's the difference between the direct contact and the indirect transmission. But what will the body do if a contact, there is a pathogen entering the body? The body is going to start its defense mechanism. And we have a lot of them. The defense mechanisms or the body defenses, here the human body has many natural defenses against pathogens. We have, for example, mechanical barriers. And these mechanical barriers, like, for example, the nose. The nose contains inside hair in the nostrils that will trap dust and germs by the hair inside the nostrils is going to trap dust and to trap, of course, germs or pathogens.
Also, number 2 can be mucus.
Yes, of course, of course. And the mucus is going to trap. Yani if the pathogens and dust escaped, because why do I say dust? Because the dust is full of germs. If the dust and pathogens escape the hair, they are going to be trapped by the mucus. Of course, they can escape from the mucus. They are going to be trapped in the trachea and so on. Yani airway passages in the humans, they have several defense stations. If the pathogens escaped one station, they are going to be trapped in another in order to allow only clean air to enter the lungs. But there are some pathogens that can, in spite of all these mucus stations, some pathogens can escape as well. The second large mechanical barrier is the skin. Now, why do I say large? Because it covers the whole surface of the body. Here, the skin has an outer layer of dead cells. And these dead cells, although they are dead, but they have a function because they contain keratin. And this keratin is a kind of protein that kills or prevents the bacteria or the pathogens from invading through the skin. Also, the skin secretes the sebum, which is an oily material that contains chemicals to kill pathogens. Also, on the skin, there are normal bacteria living on the skin normally, and they are unharmful bacteria. But when there are harmful pathogens trying to invade the skin, these bacteria are going to compete with them, and the bacteria of the skin are going to win the battle with the pathogens. All these are mechanical barriers that prevent the pathogens from entering the body. Now, suppose pathogens succeeded in entering the body, they are met with the chemical barriers. Chemical barriers means that there are some chemicals that are found in the body that will defend the body against the pathogens, and one of them I just mentioned, the mucus. This is a chemical, of course, and of course the mucus here, we have to mention it with the chemical barrier, not with the mechanical. And also the in the stomach there is hydrochloric acid. This hydrochloric acid is highly acidic, it's highly concentrated, yes, and of course it's going to kill the pathogens that are found in the foods and our contaminated food and drinks. Now again, if the bacteria or the pathogens escape these, so they are met with the last defense mechanism, which is the cellular barrier. And I just explained them. Cellular barrier means the white blood cells. So if the pathogens manage to get through all these defenses, so they are destroyed by the white blood cells, by phagocytosis, and of course by production of antibodies. These are all the barriers or the defense mechanisms of the body, and these are natural defense mechanisms. Are there artificial defense mechanisms? Yes, of course, which is vaccination, and I'm going to include it now when I explain immunity and the immune system. So let's go now to the immune system. This immune system is a system that's responsible for production of antibodies. So what is an antibody? This will produce an antibody. Antibodies are protein molecules that can fit with another molecule. So what does this mean? Suppose we have an antibody looking like this. So it can fit by this side into a complementary or an opposite shape. What is this opposite shape? Suppose I have here a bacterium, and on the cell membrane on the bacterium there is a certain protein which is called an antigen. So the antigen, if it has a complementary side shape or an opposite shape to the antibody, so the antibody, exactly like what happens in enzymes, the antibody is going to be fixed like this because it has a complementary shape. So the antibodies, they can fit into the antigens of a pathogen. Of course, after this fitting, so the pathogen is going to be destroyed, and of course the antibodies must be made exactly the right shape of the antigen. If this antigen is specific for this bacteria, what about the rest of the bacteria? Every bacterium, every virus, every kind of organism has a certain shape for its antigen. Do we have antigens? Yes, of course. Every cell has its own shape of antigens. So what about the antibodies? How are they going to fit all these antigens of different shapes? It means that the antibodies that are released in millions of forms or of shapes, because every shape, it has to fit in one and only one antigen. Yani suppose I give you an example by the shape of this antigen and antibody. There is another pathogen that has an antigen looking like this. So its antibody is going to be complementary to this shape, looking like this. Okay, another pathogen has an antigen that looks like this. So the antibody will have the opposite shape immediately, like this. So it's going to fit exactly like the lock and key of the enzymes. They are going to fit together, and then this pathogen is going to be destroyed. فالantibodies lock on the antigens, leading to destruction of the pathogen completely. And once there is locking of the antibody on the pathogen, so on the antigen, so the whole pathogen is going to be destroyed. This is one way of action of the antibodies, by fitting on the pathogen, so this will destroy the pathogen. But there is another way the antibodies act. When they fit into a pathogen, they can mark the pathogen, as if they are marking the pathogens, or they are labeling the pathogen to allow a phagocyte to come and phagocytose it. But what does this mean? Suppose I have a pathogen like this, and this is its antigen, and here came an antibody attached to it. So this attachment will allow the phagocyte, the other kind, or the kind of white blood cell, to come quickly, because it will recognize this marking or this labeling. So the white blood cell is going to come and phagocytes or phagocytose the whole antigen or the whole pathogen together with its antibody. يعني they are going to be taken inside the cell by phagocytosis that I just explained here, and the pathogen is going to be destroyed. يعني here I mentioned only a pathogen or a foreign body. But if this foreign body is labeled with an antibody, so both of them are going to be destroyed. This is how the antibody works, either by destroying the whole pathogen or labeling the pathogen so the phagocytes can come and destroy it. Yani in all conditions, the pathogen will be destroyed. If a pathogen, yes.
The antigen,
Yes.
specific shape, right, for the bacteria.
Not only for bacteria, any cell, whether bacterial, viral, of course the virus is not a cell. What I mean that an antigen is found on every foreign body or pathogen, or even our cells, they have their own pathogen, they have their own antigens. Every cell has a certain antigen on its cell membrane. This is very important طبعا for immunity, that the antibodies can come and join with them or fit into them.
So depending on the bacteria that has entered the system, that antigen will do the job.
Yes, of course. The
Antigen.
kind of specific antibody will come to this antigen.
Oh, because bacteria, they target specific places, right? Like some bacteria, like they target like maybe the lungs or whatever. So the antigens of the lungs will attack that one?
Yes,
of the shape.
the antibody, you mean. The lymphocytes are going to go through the lung to produce antibodies, and these antibodies will attack this particular antigen. We know, you look here, I told you that there are millions of forms, or in other words, millions of different shapes of antibodies. Each shape is specific for the shape of a certain antigen. It means that not only one antibody will be, one kind of antibody can destroy all the pathogens. No. For every pathogen, it has its own antibody. Why? Because for every pathogen, there is a certain shape for an antigen. As you said now, that the antibody that is specific for the shape of the antigen is going to attack not any other antibody. As I said before, that the white blood cells that produce the antibodies, they are called the lymphocytes. Lymphocytes will increase so much in number if a pathogen entered the body, and if a pathogen escaped all the defense mechanisms and entered the body, it's going to meet a huge amount of lymphocytes. And of course, these lymphocytes are going to recognize the pathogen and start to produce the antibodies specific to this antigen or to the pathogens. Specific antibodies. First of all, the lymphocyte will recognize which antigen is this. And then after recognition, they are going to produce antibodies specific for the shape of the antigen on this pathogen. This is all natural things that happen in the body to give us natural immunity. So if we are invaded by a pathogen and our immune system starts to work, of course, this is something natural. Natural immunity can be due to active infection. So we say that this is natural active immunity, where if a pathogen entered the body, or there must be a pathogen that entered the body, so the body or the lymphocytes will produce antibodies against them. There is another type of natural immunity which is called passive natural immunity. Now, what's the meaning of the difference between active and passive? Active means that there is a pathogen that, excuse me?
Isn't like active already in your system and passive is getting it from someone else?
Yes, that's it. Yes. Active means that the body is actively doing or forming antibodies, but passive, it means that the antibodies are introduced from somebody to another one if this somebody was infected. Like, the best example, of course, is from the mother to her infant.
Yes, through the milk.
Yes, of course, through the milk. Now, what happened when the mother, during her life, she was infected with pathogens. So her body started to produce antibodies inside the body of the mother. So during lactation, these antibodies will be transferred to the infant. This is called passive natural immunity. Why natural? Because the antibodies were naturally formed in the body of the mother. But there is another kind of immunity, which is called artificial immunity. Of course, this again can be active, artificial, and can be passive artificial. What's the difference between both? Active is vaccination. Now, what is vaccination? It means that I am introducing a pathogen inside the body in order to trigger the formation of antibodies. What about these pathogens? What's this vaccine or the vaccines? This is something, of course, everybody is going to think it is harmful. No, they introduce either dead pathogens or inactive pathogens, we call them attenuated pathogens, or we can introduce the toxin of some pathogens, but of course, after removing of the harmful part, that's why it's not a toxin, it is called a toxoid. All these, they act as foreign bodies that will induce the formation of antibodies inside the person who received these vaccines. It means that the vaccines, we are introducing the pathogens or the toxins of the pathogen inside the body to stimulate the immune system to produce antibodies. Well, antibodies here, they are, of course, also they are not only produced, they also produce, the body also produces something called the memory cells. فالmemory cells, they, from their name, these are cells that remember. Remember what? Remember this kind of pathogen that was introduced to the body. So if this pathogen tried to enter the body, these memory cells immediately will produce antibodies to combat this pathogen. For example, if somebody was vaccinated against a disease called measles. Measles is a disease that is caused by a virus. So if this somebody is vaccinated against measles, so the body of this person will produce memory cells. And these memory cells will remain in the blood. If a measles virus tries to enter, so the memory cells immediately will be activated, producing antibodies to destroy completely the virus before it tries to gain access to any part of the body. This is the importance or the effectiveness of vaccines. That's why we usually give vaccines to infants or to young children to protect them against diseases, to allow their immune system to form memory cells to be ready for any invasion by such bacteria or vaccines, bacteria or pathogens. But I would like to mention something here about the passive immunity that is gained from the mother, gained by the infant from the mother. Yes, it's short lasting. It has to last maximum from 6 to 8 months. طب why بقى? Because during this period, after birth of the infant, the immune system of the infant is not yet developed, completely developed. Yes, it's not ready to form its own antibodies. That's why immediately after 6 to 8 months, this infant starts to catch diseases, and the immune system is attacking them, of course. Now, but about the passive artificial, it means that we take antibodies already made in any organism, and it's not a must to be a human. We take already formed antibodies and they are injected. But when do we use this passive artificial immunity? For example, if there is a certain disease that affects, like for example, this pandemic that we are suffering from, this COVID-19, they started to take the antibodies that are formed in the bodies of the people that were infected or that were sick, the people that recovered. Their bodies have antibodies against COVID-19. So they extracted the antibodies from their blood, and they started to inject them readily made antibodies in order to attack the virus. But of course, this is, yes, it must be a successful procedure, but COVID-19 is very cunning. It changed its antigen. That's why the antibodies are no longer effective against COVID-19. Right. There is another example. For example, if somebody was moving in the street, in a garden, whatever, and their skin was injured by a nail, a piece of glass, whatever, these people, or this injured person, has to hurry and go to a hospital to take this artificial passive immunity, or to take a what we call a serum, yani antibodies already made. These antibodies already made, and they are packed in like vials like this, or ampoules. These ampoules are broken, and they take the antibody preserved طبعا in a certain chemical, and they inject it in this person. They are introducing already formed antibodies to immediately act, because if somebody is injured in the street, for example, they might catch a very aggressive kind of bacteria that leads to a disease called tetanus. And this tetanus is fatal. So that's why I said they have to hurry to a hospital to be injected by antibodies against this tetanus, because if the body waited for production of antibodies against this kind of bacteria, the body will die. Because you know that you just said that production of antibodies will take time, about 1 or 2 weeks to have enough antibodies against this bacteria. Of course, the body cannot wait. This is, as I told you, an aggressive bacterium that kills after few hours even. That's why this person has to immediately go to the hospital to take the antibodies in a form of injection. And by the way, the antibodies, as I told you, they can be prepared in any organism. It's not a must to be in a human. Yani there is a very famous way to prepare antibodies in horses. And then the horse's blood, or they take the blood of the horse and they extract the antibodies from it, and pack the antibodies in vials or in ampoules to be used taban as immediate action towards some kinds of pathogens. طيب. Of course, I just explained everything dealing with pathogens and immune system, and that the pathogens, they attack the foreign, the antibodies, they attack the foreign antigens. There is a certain type of disorder in some people which is called autoimmune disease. They are a collection of diseases, by the way, diseases. Now, what are these? I just said that an antibody or the immune system will attack the foreign cells, or because of the foreign antigens. But here in these diseases, the immune system is defective. It starts to attack the cells of the same body. Yani, for example, if I have an autoimmune disease, my immune system will attack my cells. And this is, of course, very dangerous. One of these autoimmune diseases is type 1 diabetes. Now, what is this? Here we know that the pancreas is responsible for producing or secreting, yes, insulin. And of course, insulin is produced from certain cells in the pancreas that are called the islets of Langerhans. Yani the type of the cells, اللي هو type of Islets of Langerhans, you don't have to know them. We have alpha and beta. No, you don't have to know this. You just have to know the name of these cells, islets of Langerhans. They produce insulin in a normal person. If this person is having an autoimmune disease, these cells, the islets of Langerhans, are going to be attacked and destroyed by the immune system. This will lead to, no insulin is going to be produced, so this will result in this type of diabetes. And we say that these patients, they are insulin dependent. But what does this mean? It means that they depend on insulin or artificial insulin to stay alive. This kind of disorder frequently appears in childhood. That's why we sometimes call it the juvenile, of course juvenile means children, diabetes, or juvenile onset diabetes. Always, when it happens in children, and these children, they must have an injection of insulin for life in the morning and sometimes morning and night according to the condition. And of course, these patients, they are not only receiving insulin injections, they have to take good care of their diet. Do not include too much carbohydrates in the diet, because we knew that carbohydrates are going to change into glucose. And of course, there is no insulin to allow the cells to use up this glucose. So this will lead to high levels of glucose in blood. That's why these patients, they have to reduce as much as possible carbohydrates in their blood, in the diet.